So this is our sarcomere structure. So over on this side, we have our I band, also called the light band. This giant area right here is called the sarcomere. You have your A band right here. All of these black lines that are separating the three are called D bands. So you have your actin here, and when muscle contracts, these two sides here with the red arrows are going to constrict together. So now we have our myofibro, which is our muscle cell. So just like before, between our Z lines, we have our sarcomere. These thin little pink lines here represent our thin filament, and these big ones are thick filament. So the difference between actin and myosin. So we have our muscles here. Now for a muscle to actually contract, the motor neuron, which is this giant black thing right here, it synapses with the sarcoplasmic reticulum. So when it sends a signal of contraction, the signal to the sarcoplasmic reticulum is going to release calcium ions because the membrane of the muscle depolarizes thanks to an influx of positively charged ions. When a muscle is relaxed, the actin binding sites are covered by tropomyosin in the green and troponin in the black. So, with the release of calcium ions, they bind with the troponin and the tropomyosin,
that can develop into bigger ones. So what little ones do you have?
which leaves the actin binding sites completely clear. The myosin and the actin heads are going to form together using ATP to form what's called an actin-myosin bridge. Now because of this, the myosin heads are going to swivel toward the center of the sarcomere, cocking it, getting it ready for the power stroke. But the ATP is then hydrolyzed into free phosphates and ATP, or ADP. So the cycle continues until calcium ions are pumped back into the sarcoplasmic reticulum, and the hydrolysis of ATP breaks the acetaminophen bridge, detaching the myosin from its power stroke. Now tropomyosin will go back in to block the actin binding sites along with troposin, and the muscle fibers relax.